Hi everyone, and welcome to the 12th TCIA Centre seminar of the academic year. So, my name is Adam Shepherd, and I'm also joined today by Jetji as my co-host. We're both researchers here at the TCIA Centre, and for those that are new to our seminars, we invite researchers and leaders in the areas of computational pathology and beyond. And today it's a real pleasure to introduce Dr. Johannes Lotz. Johannes Lotz is leading the research area digital biomarkers and computational pathology at the Fraunhofer Institute for Digital Medicine, MEVIS in Germany, where he works on AI-driven approaches to digital pathology, including foundation models for medical imaging and multi-stain image registration. Johannes also serves as co-chair of the working group on digital pathology of the German Society of Pathology. The team at Fraunhofer MEVIS has achieved top ranking in multiple MICCAI challenges and is driven by the goal to translate research into real-world clinical applications. And the title of Johannes' talk today is Supervised Foundation Models for Few-Shot Medical Image Analysis. So again, thank you very much for joining us. And with that, I'll pass over to you.
Yeah, thank you, Adam. So it's always a pleasure to be with you virtually. As Adam just said, the talk is titled Supervised Foundation Models for Few-Shot Medical Image Analysis. I'm Johannes, and I'm going to talk about foundation models and how they can increase robustness, especially in data-scarce situations. And yeah, we will just jump right in. If you have any questions, just let me know, and we will clarify right away. Here we are. So just briefly about what we are doing in computational pathology and digital biomarkers at Fraunhofer MEVIS. So Fraunhofer MEVIS is the Fraunhofer Institute for Digital Medicine. We do research in many different areas of medical image computing, but my focus is on computational pathology and digital biomarkers. As you may know, Fraunhofer is a non-profit research organization. We do applied research with the goal to bring research into clinical practice. And now I said computational pathology, so we're doing a lot. So actually, I started at Fraunhofer with the topic of image registration. So we went from, and maybe I can make this video. Here we are. So image registration either for reconstructing 3D volumes of tissue sections or bringing different tissue stainings together. And we recently did a study with the university hospital in Erlangen and showed that we can save about 1 minute of time per diagnostic case in breast and prostate cancer by using image registration basically as a navigation tool. So this is also a work of Nick and Daniel from our team. Otherwise, we have different projects around image analysis for histopathology, for example, here in cytology, where we Henning Höfener and Farina Koch of our team tried to or managed to classify cells in cytology into a number of different classes. And in order to bring this into bring this closer to the actual clinical routine, we're working on a smartphone app that can be attached to the microscope in order to make this easier accessible. Last but not least, biomarker discovery is an important topic. We built a web application. I will say more about this in a few slides. Okay, I won't explain how pathology works to you because I think everybody here is definitely aware of. But when we say AI and pathology, then the goal is always to have a robust deep learning model for clinical research. So what do we mean by robust? That means it should not only work on the training data you used to build the model, but also out there under real-world conditions. And yeah, the key to robustness is a diverse set of training data. So here we have in the image, we have unseen image data that goes into our deep learning model, and we have labels, maybe a patient diagnosis or stratification that we want to learn. And our model here consists of two parts, and we will get to why there are two parts. just in a moment. Typically, robustness in deep learning models is assured or aimed for by training with large and diverse data sets. And this diversity is achieved by collecting large cohorts, multi-center training data sets. And classical deep learning approach, all my many and diverse training data sets must be problem specific. So this is easy in some cases, but difficult in others, for example, in rare diseases or in personalized medicine. So this is how it looks like. So we have our soon-to-be robust deep learning data set. We have like training data from different hospitals all over the world. We have our deep learning model that is trained, and we have all these labels attached to the images that makes the model understand what we wanted to learn. Here, for example, it's Gleason grading. And for Gleason, this is very simple. Well, not very simple, but rather simple because this is very frequent and often diagnosed. But what would we do if we have like only rare disease or fine-grained subtypes as in personalized medicine? Then this is the problem to gather this large diverse set of training data. And foundation models are one approach to make this problem a bit easier. So what foundation models do is to incorporate data diversity. So if you look at how a human learns, so radiologists, pathologists, they do not start from scratch for every organ. So they learn a basic medical training. And that is exactly, well, maybe not exactly, but this is somehow what foundation models do. So foundation model is a model that is trained on broad data and can be adapted to a wide range of downstream tasks. So this means that we can, in the model already, incorporate these large amounts of data and the data diversity, and then maybe later adapt them to our prostate cancer grading problem, for example. So we get to the training part in a minute, but let's just imagine we managed to train a foundation model. So this is the left part of our deep learning network with diverse training data. And then what we can do is we can freeze the model. So now our model is frozen. We can then put an image patch, an unseen image patch, or an already, so it doesn't matter, any image patch into the model, and we will get a vector of image features or maybe a tissue or a patch signature for this specific image patch. So the goal of the training is to pre-learn or pre-train the patterns that we need or the tissue features that we need later to solve so-called downstream tasks. So that's how it looks like. So we now have our frozen model. We put our image patch in. We still have our label for our Gleason grading, for example. And then the only thing we need now to do is to train a very small part of the model. So this is the so-called downstream task. So here, this part is frozen, this part is trained. And since this is very small, we only need so the training is very cheap, and we only need very little, or we can do it with very little data, or at least with less data, and everyone is happy. So the only problem is the high training cost of the foundation models. So this is for large language models, which is different from what we do in pathology, but still to get an idea about how expensive it is to train these models. So this is the estimated training cost of select large language models from a 2024 report. I didn't find any newer data. So, but already then we have like $200 million for Gemini Ultra in 2023. So this is very expensive, and one also has to think about that the training is not a one-time thing. So once you are done training your model, you start again to make it better. So this is really a cost that one needs to take into account. In pathology, the models are a bit smaller, and we don't need like to train with the whole internet and any source we can find. So we are a bit more restricted in training data and also model size. Also, the application is much more narrow. So in pathology, the current foundation models seem to be at least, so the training cost seems to be at least in the tens of thousands of euros. So as a ballpark number, so training one foundation model like this here produces as much CO2 emissions as one flight one flight per person from New York and back. Or the electricity consumptions of each of your households on average. So so when we started to work on this topic of foundation models like years ago, we came from a bit of a different angle. So the term foundation model was not coined yet, and we were looking for an AI-based model to start our developments from. So something that has somehow pre-learned important general concepts. And we were coming from ImageNet as everyone else as pre-trained model, and we were looking for something more pathology specific. So we wanted to learn these pathology concepts and aim to use like a supervised approach. We call the model tissue concepts. And in order to distinguish the self-supervised and the supervised approach, one has to like go one step back and look at the self-supervised training approach. Most of the foundation models today have been trained using a training scheme similar to DynoV2, which is a combination of masked autoencoder and contrastive learning. So basically what the model needs to do is we mask parts of the images and parts of the latent space of the model. The model has to like reconstruct the image, and at the same time has to produce consistent features under image augmentation. So we have a student and a teacher model, and one of them only gets a full image, and the other gets like augmented and cropped images, and both need to kind of end up with the same representations or signatures or features or however you may name it. And what we see here is that at this using the self-supervised approach, there is no medical background or medical details. So there's nothing about the patient in there. It's just about tissue structure. So the model can only learn the intrinsic tissue structure of the training data. And if you think of how maybe a medical student learns, this is a completely different approach. So they learn how a cell looks like and will recognize abnormal shapes based on explanations in textbooks. So in the supervised learning approach, it's more like this textbook approach. So each image has some kind of label, and the goal is to learn like medical concepts in the tissue. So we, as I said, call the model tissue concepts. We don't use the self-supervised approach, we use the supervised approach. This is mostly work of Till Nickel and Raphael Schäfer, who might be even in the call. Not sure. If you are, you can wave. Not sure. What we propose instead is supervised training. So based on examples, what a tumor looks like, for example. And what we do is like a multitask training scheme with now over 60 tasks that the model has to learn at the same time. So we have a base model, we have all these different tasks, and we put them through the model. And we have the labels for all of the tasks. It can be like segmentation labels or patch classification labels, slide classification labels, object detections, and from a number of different tasks. And we have a bottleneck in the model somewhere here. We will get into the architecture a bit deeper in a few minutes, but for now we have, let's just say we have this bottleneck in here. And the features or representations that the model learns have to work for all of these tasks. So if I put in mitosis image, then the features that we have that we learn in the model have to work for mitosis detection, and same for all the other tasks. So we're learning something, universal. That's the goal. And the advantage is that due to this large shared part, the shared encoder, all the different tasks like take advantage of each other, or so there's some synergy between the tasks. And if I need information from one task, I can reuse it in another task, and in the end maybe I can be even better as if I only opposed to if I only learn one task at a time. Yeah, we started with a small, relatively small model, so based on a Swin transformer with size tiny, and then also trained a few larger variants. And then we had a look at the performance and also at the training cost of this model. And what we see here is on the left side, we see different models. In blue are two tissue concepts variants, one with the Swin transformer, the other with a convolutional approach. And then we see some other self-supervised models, and on very light green, the ImageNet base model. And we compared it on 5 different tasks and measured the accuracy in each of them. And what we found out is that the supervised approach on average gets the same accuracy or the same performance than the other models. But when we look at the training cost and the number of images needed, then we see that we only need like 1% of the training time. And yeah, depending on what you compare, 5 to 15% of the training data. It really depends on the actual training approach and what you compare the model against. And it's also not so easy to compare because it's often not reported how expensive training is. Okay, and so now let's have a bit deeper dive into the model architecture. So this is the same as this here, right? So same thing, but in different visualization. So what we have here is we start from here. We have our images that go into the joined into the image encoder. Then we have different paths we can go. So we have for patch segmentation, there's a decoder. We have, what is called, it's just a linear layer, what's called a squeezer for patch classification. And we have some context transformer to, if we want to have 3D images, then we need to put the slices of the 3D image into context, or if we have a whole slide image, then we need to put all the patches into context, and this is what the context transformer does. And yeah, in the end, this here on the left is the part that will be frozen. So the frozen part of the foundation model. And you can configure it how you want, but that's how we currently do it. If you now have like the pre-trained foundation model and want to apply it to a patch segmentation task, then you retrain like the decoder and use it for your patch segmentation, but the decoder only takes like 4% of the overall weights of the joint model. So the largest part, more than 90%, is this encoder. Yeah, and image patches are like the smallest units, the atoms that we use in the training approach. Everything goes down at some point to an image patch. Yeah, one application that we did with this approach is survival analysis. So what we did is we pre-trained the model. We prepared it for prostate cancer survival analysis. We pre-trained in a project, publicly funded pro-survival project with university hospitals in Berlin and Frankfurt, and the office, fine for informatic, not sure how to translate it, here in Oldenburg. And together we did a project, and we pre-trained the model with prostate-specific tasks. So we did a Gleason training as pre-training, and we used some PIN4 stains, for example, put it also in the pre-training. And then in the end did a survival training. So instead of a Gleason grading, we had survival labels for each of our patients, and we trained the model. And that what you can then do is to look at the attention, of course, and see where the model or which patches in the images were important for the actual survival decision. And we then took all these images, so the high attention images, and showed them to pathologists and let them rate on, I think, a 1 to 5 scale on aggressiveness, infiltration, cribriform patterns, mitosis, inflammation, on these five criteria for all our high importance patches. And we made a distinction in two classes. We had patients with biochemical recurrence in orange, and we had patients without biochemical recurrence in pink or purple. And the key here is that we only used patients of the grade group 3+3. So those patients, if you only look at Gleason grades, then all those have like a similar survival prediction. But we looked into the images. Can't we like make a survival prediction of these otherwise very homogeneous patient group and see, is there more in there that distinguishes these patients? And what we find is that on most of the criteria, there is not much of a difference, but we can see a difference that the patients with biochemical recurrence had a much higher score on cryptiform patterns and the lower score on inflammation. And this is very interesting because if you now look in the guidelines, you see that, okay, yeah, cryptiform patterns should not be in grade group 3+3. So actually, like 10 years ago or so, I forgot when exactly, the grading was changed. And if you see cryptiform patterns, this shouldn't be in your 3+3 group anymore. But since our data is like much older than 10 years, we of course had still like patients with cryptiform patterns in this grade group, and the model was actually able to detect these patterns and find that they are associated to a higher or to a lower chance of survival. So in the end, we didn't find new medical information or knowledge, but at least we could verify that this approach kind of works. Yeah, and so what we want to do now is to see if we can actually use this approach. We have just a project starting next week where we want to evaluate this in other diseases and see if we can like train this model end to end with patient level labels and see if we can like find new knowledge in the tissue by looking at these attention scores, for example. And if you have some similar problem, we would be very happy to look at this together. Yeah, then Adam asked me if we could talk about the Unicorn Challenge. So the Unicorn Challenge is the MICCAI challenge called the Unified Benchmark for Imaging in Computational Pathology, Radiology, and Natural Language, which naturally comes to the abbreviation UNICORN. And the goal in the challenge was to find robust and general foundation models. And the task was on, I think, 12 or so different tasks with very few training data, adapt a pre-trained foundation model to the specific task. So I think there were like 48 patches per task or so. So from classification of histology images, also in radiology, classification of malignancy of lung nodules, finding tumors in MRI, segmenting other structures, spine MRI, for example. And yeah, so we participated in this challenge, and our model performed very well. So we made the first place in radiology and pathology and also overall. And yeah, and one attribute or one feature of the supervised approach is, of course, that you can prepare the model to a specific task. So what we can do, so the organizers of the Unicorn Challenge provided a list of resources with tasks that might be similar to what was actually done in the challenge. And we, and maybe many others as well of the participants, used, of course, this data to pre-train the foundation model to the specific task. So we added a few tasks to the foundation model prior to sending it in for the challenge. So in the base model of tissue concepts or UMED-PT, we are not very stringent in naming this thing, was trained with like 60 tasks, starting from very general total segmentator, ImageNet 21K, but also Panda, or there's something called RUD ImageNet. So whatever we could find on public data, we put into the pre-training. And then we extended this towards the UNICORN challenge. And then what we can do, we can use these different arms of the pre-training mechanism also for the inference basically, or for the few-shot learning. So for example, for the pathology tasks with slide level labels, we extracted like 10,000 patches from every data set with given tissue mass. We used level 0 for biopsy data, otherwise 1 and 0. And then we use the transformer layer here to find these patches with the highest attention, and then only took the 1,000 best patches, put them through again, and trained them with the actual task labels provided as a challenge. And concatenated a few variants of the same representations to solve the actual tasks. Similarly, for the pixel level tasks, so we have dense features that can be used as patch representation. So basically, for every pixel, we have like 32 channels that we can extract from the original tissue patch, and these can then also be used, to adapt to a downstream task with few training data. Yeah, we not only used pathology data, but also the same model for radiology. So what I didn't show before, the model is actually also trained with radiology data, so as part of the 60 tasks. So we can't prove that this helps to have the radiology data at the same time, but it doesn't seem to hurt either. So since MEVIS as an institute is not only interested in pathology, but also in radiology, it made sense for us to put both into the same model if we start to train something like this. Yeah, so what's next? So what we're currently working on is a risk modeling web application. So it's still work in progress, but it's the same thing that I showed earlier. So you can like put in any data with patient level labels and have more or less an interactive approach to train your model. So to make it easier for non-technical people to train these models and look at the data and see if these attention scores provide some useful information. So this is still in the very early shoes, but early shoes, maybe not an English wording, but so very early in development. But that's one way we can think about using this later on. This is on the software side, mostly done by my colleague Nick Weiss. And we are looking for more applications to apply this to new medical tasks. So if you have, as I said earlier, if you have something similar, we would be very happy to give this a shot. And something else that has been done or developed many times is a few-shot segmentation. So what you can also do relatively easy is to just take some tissue, make a few annotations, and it's really astonishing. So this model has seen colon tissue, right? So this is maybe not that astonishing, but you can really get very good results with just a few training examples, some glands and some background. And you can train this really quick because you only need to train like a very thin layer of model. Makes sense to train at the boundaries of different structures. So this I increased the overall video by factor 1.5, but otherwise I didn't speed anything up here right now. And then we have already a trained model. We can apply it to like an arbitrary region of interest and see this is already quite okay. And then you can like, of course, interactively refine the model and say, okay, well, here this should be background. We want to separate the glands a bit better, and and so on and so on. So this can then be retrained, and in the end, looks like this. So I think so. Yeah, to me, this is always quite astonishing how well this works. Yeah, over time we developed like a small model zoo. So we started with the, which is currently, if you go to our GitHub page, we have the default model, which was the model that is originally presented in the original paper. We then had a more pathology-focused model. Then we have the model that was used in the unicorn encoder, in the unicorn challenge. And the latest one is a project of Till Nicke, where we just trained the model on whole slide level labels. So there, in the training of this model, no patch classification labels have been used. It's purely trained with labels from diagnosis, from reports, from so whole slide level. So each patient, one label. And turns out that you can also use this for a model training, which is pretty neat because we see now that there is still some unused data out there. But if you want to train with patch level labels, then there's a limited set of labels that you can use. And if you extend to whole slide level labels, then of course so every pathology report has whole slide level labels. So you can use them also for training this kind of foundation model. Yeah, so we would be very happy if you gave us a try. So the model is publicly available. The papers are available. You can just download it from our GitHub page and use it, and we would be happy to hear from you. Or if there's any problem or something does not work out, this would be also very interesting. One can, of course, also add data, always add data sets to the pre-training if some specific. So we see that the out of domain performance is not bad, but can always be improved by adding some specific data set similar to the target problem. So this would be very interesting to hear from you if you want to use this. And yeah, I want to thank you now for your attention. And if there are any question, I would be happy to answer them.
Amazing. Thank you very much, Johannes, for a really, really interesting talk there. And yeah, congratulations again for such a high performance on the Unicorn Challenge from your team. So yeah, let's open the floor to questions. Does anyone have any questions they'd like to kick us off with, either online or in person? I can. Sorry, actually.
Hello, may I ask a question?
Yeah, of course.
Hi, Ihor is my name. I am a resident pathologist at Heidelberg. I have some beginner level of experience with computational biology, and thanks a lot for your talk. I have two questions. First, maybe can you give some short ideas or advices to someone like me who is actually doing all these annotations? So I worked as well with foundation models and convolutional networks. I have no clue how they are different from each other from the informatics point of view, but maybe you can give some advices. How should be the annotations done? Should I rather annotate big areas and more annotations, or rather, as you just showed, few-shot learning, for which models are such few-shot learnings well, and rather small structures? With example, like if I have some tumors with not maybe necessarily gland formation, but some solid areas, should I also include this trauma, or should I really draw each cell or very small groups? And how is it different between foundation model and convolutional networks? Maybe you could give some brief ideas or direction where to think about.
Yes, Raphael, maybe he can also comment. So maybe first part of an answer. So I'm not sure how the difference is between, like, if there's a difference between how you do it for a foundation model, or if there's if it's a foundation model. So what we have seen, which works very well, is to do it interactively with the model in the background, and to see where the model still needs additional training labels, and then walk yourself through. But maybe Raphael wants to add something to this.
I wanted to say exactly the same.
Excellent.
Then it seems that you are talking truth if you are both saying the same. And the second question might be a bit shorter. We have here at Institute MICCAI software, also from Fraunhofer Institute. How is it different from your RISMO program, what you just showed? Is it some similar stuff or different?
No, it's rather different. So the Fraunhofer Institutes themselves are relatively independent. So the only thing that you get in the MICCAI software from us is the image registration, which is in there in the newest version. But our foundation models are not yet in there. And they are using, so in MICCAI, there's a slightly different approach implemented where, so it's, yeah, I'm not sure how much I can really say about it, but it's more like a clustering-based approach as opposed to our straightforward fine-tuning approach, as far as I know. But maybe you would need to ask them to see what's actually implemented.
Okay, thank you for the answers.
Fantastic, and also we're on this particular slide, Johannes. So is this RISMO software? Is it available? How's the? Because I don't think I've seen it before, but it looks like a really good piece of software.
Yeah, it's not yet available because we are still working on it. And I think so. What we will try to do is to make it available as a web application at some point in the future. But there's still too much left to do to release it just yet. So not yet, but maybe soon.
Sure, thank you. And also a question, I suppose, on the foundation. Sorry, the model you had for the Unicorn Challenge. Do you think? I know you said you hadn't trained it separately, but I was wondering what you thought in terms of training these encoders with both radiology and pathology data. Is it sort of a synergistic, sort of better overall model, do you think, or is it better just to completely separate them when analyzing different types of data as such?
Yeah. So my original impression was, it doesn't. There's not much synergy, but it's difficult to say. And I know that at least Raphael had a different opinion on this. I'm not sure if he wants to say it, but we discussed this a lot, but I'm not sure if we have a definite answer.
Sure.
Getting this answer is definitely on the roadmap. But it's quite expensive to really answer the question. I think so. We would have to train another model from scratch, only using part of the data or the parts we're interested in, for example, pathology versus radiology. So it didn't happen yet. But
That
what we
makes
can
sense.
definitely say, it doesn't hurt. So the same model can learn two things.
No, brilliant. And I think, Jack, did you say you had a question you wanted to ask?
Thank you for your presentation and also congrats on the challenge. So I would like to hear your view on this, on foundation model. So I mean,
much better.
Okay, that's good. So since your model is trained using a supervised task-specific, also multitask kind of learning objective, do you think it still fits within the scope definition of foundation model? In other words, in your opinion, what is the definition of foundation model? Is it defined by the training paradigm, or is it defined by the features that is learned, or is it defined by the size of the data, or just
Yeah, so I mean, if you ask me, obviously, I would say it depends on what it can do, and not what's in there or how it was trained. So I would stick with this definition. I liked it. Actually, made it also its way to Wikipedia at some point. So trained on broad data generally using self-supervision, okay, there I might not be on board, but can be adapted to a wide range of downstream tasks, and so to build the foundation of some other model. So that's what I would say. But this is really, that's semantics, right? So if how you want to call it. I think so nowadays, I think the term foundation model is used rather inflationary. So everything is a foundation model now. So in this definition, this is of course also a foundation model, but yeah, really depends where you want to draw this line. I don't know.
Yeah, it's pretty hard to define, I guess. Also, just one more question. I think you mentioned it, but so how does it perform on tasks that hasn't been trained during training? Have you compared with other models, foundation models?
Yeah, maybe Till, do you want to say something about this? So this is results I took from Till's work.
Yes, of course. So I think the left plot, the rightmost bars are lung tissue, which was not present in the training of the tissue concepts model. And we can see that the performance compared to other foundation models that have seen lung tissue in their self-supervised training drops, but compared to ImageNet, which also hasn't seen any lung tissue, it performs better. So there is some transfer learning from tissue specific areas, like if you just train on like a broad range of tissue but leave one specific organ out, then you can definitely see a transfer performance, but it is not as good as if you would have this specific type of tissue in your pre-training.
Yeah, okay. Yeah, that's great. Thank you.
Brilliant. Thank you very much. Do we have any more questions from anyone online or in person? Ask any questions? No. Well, in that case, I think we'll call the seminar to a close then. But thanks again, Johannes, and thank you very much for such a really, really interesting talk. So just a quick reminder to everyone else that our next seminar is scheduled for Monday the 13th of April. We have a talk by Dr. Sahar Nasser from Emory University, and she's going to discuss her work on SAGE, an agentic framework for interpretable and clinically translatable computational pathology biomarker discovery. So hopefully see you all there. But for now, thanks again, Johannes, for a really, really great talk and for
Yeah, thank you. It was a pleasure. Yeah. Bye bye.